Today we are going to talk about the switch from acute to chronic infection in Pseudomonas aeruginosa that involves the GAC-S and GAC-A system along with RET-S. GAC-S, whose exogenous signal is unknown, is a sensor kinase that interacts with GAC-A, a response regulator. Through two internal phosphotransfer reactions, the conserved histidine residue on GAC-S transfers a phosphate group to a conserved aspartate residue in GAC-A. LAD-S is an orphan sensor kinase, which means that it has no associated response regulator. Also interacts with GAC-S but through an unknown mechanism. Then, the phosphorylated GAC-A binds to the GAC box sequence, which is minus 180 base pairs from the transcription start site of both RSMY and RSMZ. Once transcribed, RSMY and RSMZ sequesters RSMA, a post-transcriptional repressor of PEL and PSL genes. RSMYZ has stem loops, where at the end of the loops are sequences that are essentially ribosome-binding sequences, AGGA. RSMA binds to the stem loop of RSMYZ, which then does not allow RSMA to repress the translation of PEL and PSL. As a reminder, PEL and PSL genes are important for biofilm formation. RET-S, another orphan sensor kinase, also interacts with the GAC-S and GAC-A system by forming a heterodimer with GAC-S. This heterodimer is non-productive in that no activation or signal transduction through GAC-S may occur. Without GAC-S activation, RSMA is not sequestered by RSMYZ. Then, RSMA is free to repress the translation of PEL and PSL, thereby inhibiting biofilm formation. Without the formation of a biofilm, Pseudomonas aeruginosa works in acute infection mode. So how does one get chronically infected with Pseudomonas aeruginosa? Chronic infection of Pseudomonas aeruginosa involves cyclic di-GMP, or C-di-GMP. Diguanidate cyclase, also denoted by WSPR, takes 2 GTPs and makes C-di-GMP. High levels of C-di-GMP has been shown to lead to the expression of type 6 secretion system. More so, high levels of C-di-GMP is related to RET-S because it has been experimentally shown that there is an increase in C-di-GMP levels in RET-S mutants. C-di-GMP levels are also affected by a phosphodiesterase, also denoted by PA2133. The phosphodiesterase breaks down C-di-GMP, and low levels of C-di-GMP leads to the expression of type 3 secretion system. Further, in RET-S mutants, the GAC-S-GAC-A system can be activated, which in turn produces RSMYZ that sequesters RSMA. Without RSMA, there is no repression on PSL and PEL, thereby resulting to the formation of extracellular polymeric structures. This leads to the formation of biofilms and to the chronic infection mode of Pseudomonas aeruginosa. Thanks for watching!